250 meters below the hills of Saxony, Switzerland, in a tunnel system flooded with water no one should be able to survive in, something is hanging from the ceiling. Not stone, not the pale mineral fingers you'd expect in a cave. Orange-brown slime, formed into shapes that mimic stalactites, growing slower and stranger the deeper you go. On the floor, in the drainage channels, a different kind of slime moves like thick gelatin threads, clinging to the walls of a passage where the water registers a pH close to lemon juice and carries enough dissolved uranium to set off a radiation detector. This isn't a hypothetical. It's a real, studied, published environment, and what researchers found living inside it rewrote what biologists thought was possible in a place this hostile. The location is the Königstein mine, and its history explains why the conditions down there are so extreme in the first place. During the Cold War, this was one of East Germany's active uranium mines, feeding ore to the Soviet nuclear program. Getting uranium out of solid rock isn't a matter of just digging it up. Miners pumped sulfuric acid directly into the rock formations underground, dissolving the uranium out of the stone so it could be pumped back out in liquid form. That's an industrial process that, at scale, means tons of one of the most corrosive substances on the planet, injected directly into the earth, year after year, for decades. When the mine stopped being profitable after German reunification, the operation shut down, and the tunnels were flooded deliberately. A standard remediation strategy meant to keep contaminated groundwater contained rather than spreading further into the surrounding water table. What that flooding created, whether anyone intended it or not, was a sealed, stable, extraordinarily hostile aquatic environment. 250 meters underground, there is no sunlight at all, not filtered, not indirect, none. The temperature sits in a narrow, constant band, 13 to 15 degrees Celsius year-round, because at that depth, the surrounding rock insulates the water from anything happening on the surface. The water chemistry is where it gets genuinely brutal. In the drainage channels, researchers measured a pH around 2.86. Up where the slime formations hang from the ceiling, it's even more acidic, closer to 2.55. For reference, battery acid sits around 1, and stomach acid is roughly 1 to 2. This water isn't quite that extreme, but it's firmly in territory that would chemically burn exposed skin over time, and that most complex life would consider instantly and completely lethal. Layered on top of that acidity, dissolved uranium running as high as 39 to nearly 70 milligrams per liter in the ceiling formations, iron concentrations reaching into the hundreds of milligrams per liter, and sulfate levels above 2,000 milligrams per liter. This is, by every reasonable standard, one of the least hospitable liquid environments accessible to direct human study on the planet. When researchers from the Helmholtz-Zentrum Dresden-Rossendorf, led by scientist Isabel Zirnstein, went down to examine what was growing on those walls, the working assumption going in was reasonable and, on paper, well supported by everything known about extreme environments up to that point. Bacteria and archaea, the simplest forms of cellular life, are famous for tolerating conditions like this. They lack the complex internal machinery of more advanced cells, which in a strange way makes them more chemically robust, less fragile, better suited to surviving acid, heavy metals, and radiation. Complex cells, the kind with a proper nucleus holding their genetic material, the kind found in animals, plants, fungi, and much of the microscopic world beyond bacteria, were expected to be scarce to non-existent in an environment this punishing. That assumption turned out to be wrong, and not by a small margin. The two types of biofilm growing in that tunnel system aren't identical, and the differences between them tell you something about how this ecosystem is actually structured. The formations in the drainage channels, the ones behaving like flowing gelatinous streamers, are dominated by iron-oxidizing bacteria and form directly in moving water. The ceiling formations are structurally different. a solid, orange-brown base with a softer, translucent, almost gel-like layer at the growing tip. And critically, the eukaryotic diversity inside these ceiling formations increases with the age of the structure. Older stalactite-like growths host a noticeably richer mix of complex organisms than younger ones, which tells researchers this isn't a random accumulation. It's a successional process, an ecosystem that builds complexity over time, the same way a forest gets more biologically layered the longer it's left undisturbed. At the base of the entire system sits a single organism doing almost all of the actual energy production, a bacterium called Pherovum myxophaciens. This bacterium doesn't get its energy from sunlight, the way nearly every food chain you're familiar with ultimately does. It gets its energy by oxidizing dissolved iron and sulfur compounds directly out of the water, a chemical process called chemolithotrophy. In practical terms, this bacterium is eating rock chemistry and turning it into usable biological energy. And in doing so, it's building the entire base of a food web in a place that will never see a single photon of sunlight for the rest of its existence. Feeding on that bacterial base is where things get genuinely strange, because this is where the eukaryotes come in, and they don't show up as an afterthought. They show up as a dominant, structured part of the community. The single most abundant complex organism in the entire system is a tiny flagellate called Bodo-saltans, a single-celled organism that swims using a whip-like tail, and in these biofilms it accounts for somewhere between 20 and 40% of the total eukaryotic biomass. Bodo-saltans is not an exotic, rare organism in general. It's found in freshwater and marine environments across the planet. What made its presence here scientifically significant is that it had never previously been documented surviving in an environment with uranium concentrations this high. Finding it thriving here, in numbers substantial enough to dominate the biomass, was itself a genuine first. Alongside it, amoebae and a related group called Heterolobosea together make up roughly another 30% of the eukaryotic community. These include amoebae genera like Vanella and Thecamoeba, along with heterolobosean genera such as Volcampia, Naegleria, and Paravolcampia, organisms that move by extending and retracting shapeless projections of their own cell membrane, engulfing whatever they can absorb from their surroundings. Above them in the food web sit the predators, ciliates, tiny organisms covered in short hair-like structures. They use both to move and to sweep food toward themselves, including species from the genera Oxytricha and Vorticella. These ciliates aren't just filtering bacteria out of the water, they're actively preying on the flagellates and amoebae below them, meaning this system has a genuine, functioning predator tier, not just a single layer of organisms passively absorbing chemical energy. Rounding out the community are fungi, making up somewhere between 5 and 20% of the total biomass and functioning as decomposers, breaking down dead organic material and cycling it back into the system. A stramenopial species called Diplophrys arcari, another organism documented here for the first time in a uranium-rich acid mine environment. Members of the phylum Apicomplexa, a group that on Earth's surface is best known for containing parasites, represented here by organisms in the family Eimeriidae, and perhaps most surprising of all, animals. Not vertebrates, obviously, but members of the phylum Rotifera, specifically from the class Bdelloidea, microscopic multicellular animals with proper organ systems, confirmed here for the first time in an environment this contaminated with uranium. Researchers even found mites, tiny arachnids, including a species called Schwebia pseudotsugae, an organism whose relatives are far more commonly associated with compost heaps and rotting wood than with 250-meter-deep radioactive mine water. Identifying all of this wasn't done through observation alone, and it's worth understanding how the research was actually conducted, because the two methods used caught different things. The first approach was straightforward light microscopy, using phase contrast microscopes at 40 and 100 times magnification, which let researchers directly observe and physically identify organisms by their shape and movement, the ciliates, the flagellates, the amoebae, all visually confirmed. But morphology alone has limits, especially with organisms that look similar to each other or that don't survive well once removed from their native environment for closer study. So the team also ran a molecular analysis, extracting genetic material and specifically targeting a gene called 18S rDNA, a standard genetic marker used across biology to identify and differentiate eukaryotic species. They cloned and sequenced 103 individual genetic samples pulled from the biofilms, sorted them into 23 distinct genetic groupings, and compared each one against international genetic databases using a tool called BLASTON, before building out phylogenetic trees to map how everything they found related to known species. That combined approach, direct visual confirmation paired with genetic sequencing, is what allowed the team to also identify something else entirely, a cluster of 13 genetic signatures that didn't match any known, previously classified organism at all, an acid-tolerant eukaryotic group, present and active in the samples, that simply doesn't correspond to anything already in the scientific record. None of this is presented by the researchers as a curiosity for its own sake, and this is the part that gets lost when a story like this gets flattened into a single dramatic headline. The actual scientific conclusions in the published study are specific and carry real weight for how contaminated sites get managed. The researchers state plainly that eukaryotes colonize extreme habitats like this one to a far greater extent than had previously been assumed, and that these organisms likely play a substantial role in cycling carbon through acid mine drainage systems, meaning they're not passive passengers in this environment. They're actively processing and moving organic material through the system the same way earthworms or fungi do in an ordinary forest soil, just built on a completely different chemical foundation. The second conclusion is the one with real practical consequences. The presence of this much biological activity means heavy metals, including uranium itself, aren't simply behaving according to straightforward chemistry in an environment like this. Living organisms interact with, absorb, transform, and redistribute those metals as part of their normal biological processes, which means any attempt to predict how radioactive contamination will spread, settle, or persist in a decommissioned mine like this one has to account for biology, not just geochemistry and thermodynamics. The researchers say this directly. Performance assessments of contaminated sites shouldn't focus exclusively on chemical models. They need to incorporate microbially mediated reactions, because those reactions can meaningfully change how toxic heavy metals move and where they end up settling over time. In plain terms, if you're trying to model where the uranium in a flooded mine is eventually going to go, and you're only accounting for water chemistry and ignoring the fact that a functioning, multi-layered ecosystem of bacteria, flagellates, amoebae, fungi, and rotifers is actively metabolizing inside that water, your model is incomplete and possibly wrong in ways that matter. It's also worth placing Königstein alongside other extreme acidic environments studied elsewhere, because the comparison sharpens what makes this particular site unusual. Rio Tinto, in southern Spain, is one of the most heavily studied acid mine drainage environments in the world. A river turned a deep rust red by dissolved iron, with a pH in a similarly brutal range. Rio Tinto, though, sits entirely at the surface, exposed to sunlight, which means its ecosystem can include photosynthetic organisms like algae directly harvesting solar energy, on top of the same kind of chemical energy processing happening at Königstein. That access to sunlight is a large part of why Rio Tinto supports a noticeably higher diversity of species than the flooded tunnels in Saxony. Königstein's ecosystem, sealed 250 meters underground with zero light input of any kind, has to run entirely on chemical energy pulled directly out of dissolved iron and sulfur, with no photosynthetic option available to any organism there at all, ever. That a complex multi-layered food web with genuine predators and decomposers and previously undocumented species can still establish and sustain itself under those specific constraints. No light, extreme acid, high radioactive metal content, is the detail that separates this from most other extreme environment studies. There's a reason findings like this get cited well beyond the narrow field of mine remediation, and it has to do with how scientists think about where else, beyond Earth's surface, life might plausibly exist. An environment that demonstrates complex, multicellular life surviving and organizing itself into a functioning ecosystem without sunlight, powered entirely by chemical reactions between rock, water, and dissolved metal, is a working, physical example of exactly the kind of conditions astrobiologists consider when they evaluate subsurface environments on other worlds. Ice-covered moons with liquid water oceans sealed beneath their surface, or planets and moons with volcanic or hydrothermal activity providing a chemical energy source in the total absence of light. The Königstein study itself doesn't make that leap explicitly. It's a paper about mine remediation and microbial ecology, published in a journal focused on exactly that. But the underlying result, that this level of biological complexity doesn't require sunlight and doesn't stop at bacteria, is precisely the kind of evidence that gets folded into the broader, separate scientific conversation about what subsurface habitability might actually look like elsewhere in the solar system. What makes the Königstein tunnels worth remembering isn't a single organism or a single number. It's the shape of the whole thing. A producer running entirely on rock chemistry, a first tier of grazers processing that energy, a functioning predator class hunting the grazers, decomposers cycling the dead material back into the system, and, sitting quietly inside all of it, a cluster of 13 genetic signatures that don't match anything already known to science. 25 years after the mine went quiet and the tunnels filled with acid water nobody expected to support anything at all, that's what was actually waiting down there, not an empty poisoned void. A complete, functioning, ordinary-looking food web, built entirely out of the one thing everyone assumed would make life there impossible in the first place.